Let me briefly mention several other scientific areas related to genetics that, in my opinion, support the Bible and weigh against secular evolutionary theory. The waiting time problem, genetic entropy, and mitochondrial Eve and Y-chromosome Adam. So, the waiting time problem. Many people conceive of evolution as proceeding steadily forward, one mutation at a time, with each mutation conveying a slight advantage to the organism. But it turns out that generating a new gene that produces a new protein will often need several coordinated mutations to occur in the same gene at the same time. For example, scientist Michael Behe says that studies show that 2 amino acid substitutions were needed in the protein PFCRT in the malaria parasite to make the parasite resistant to chloroquine medication. Scientists Michael Behe and David Snoke ran computer simulations to investigate this and published their results. Behe and Snoke found that 2 mutations could arise quickly by chance if the population size was enormous, more than 1 trillion individuals, which is much larger than any multicellular animal species which has ever lived. Or alternatively, 2 mutations could arise by chance in a reasonable multicellular animal population size, but only after 10 billion years or more. There was one sweet spot in their simulations where 2 mutations could arise by chance after 100 million generations in a population size of 1 billion or more. However, if more than 2 mutations were needed to make the new gene and protein, their simulation showed that that is beyond the limit of what mutations can produce in the entire secular estimated age of the universe. Evolutionists Rick Durrett and Dina Schmidt performed their own simulation, and while they disagreed with Behe on the specific numbers, they confirmed that 2 mutations would take about 200 million years, and the chance of more than 2 coordinated mutations happening during the entire history of life on Earth is unlikely. Similarly, scientist John Sanford and colleagues have published a simulation to measure the waiting time problem in the hominin population, which is a much smaller population than the population of single-celled organisms like bacteria. Their simulation estimated the amount of time before 1 or more mutations would be fixed, that is, completely established throughout a population of about 10,000 apes or humans. They found that it would take an average of 84 million years to fix a string of 2 DNA nucleotides, and fixing 3 mutations would take 380 million years. 6 mutations would take more than 4 billion years, more than the evolutionists assumed age of the Earth. And fixing 8 mutations would take more than 18 billion years. So that's more than the evolutionists assumed age of the universe. You can read these papers at the links in the supplement. So if it's not possible for mutations in natural selection to generate such a small number of mutations in a row, it's very unlikely that tens of millions of nucleotides could have been generated within the evolutionary scenario of apes and humans diverging in just 6 million years. And again, the problem is not just the sheer number of needed mutations, but the problem that several specific mutations in a row would need to happen together in a coordinated way to complete the new genes to create the new proteins. The evolutionary scenario just doesn't make sense scientifically. Another issue is that it turns out our genome is gradually deteriorating as genetic mutations accumulate in our bodies over time and get passed on to our descendants. Originally, evolutionists thought that natural selection would get rid of harmful mutations and keep the good beneficial mutations. But natural selection only works on very good or very bad mutations, and most mutations are nearly neutral in their effect on fitness, although they're much more likely to be slightly harmful than slightly beneficial. This diagram from the book Genetic Entropy shows this concept, as it shows the relative proportions of harmful or beneficial mutations and the no selection zone in the middle. Natural selection cannot filter out these near neutral mutations in the middle area. Scientists have found recently that each generation inherits about 100 new mutations from parents. So all of us are mutants, and we all have about 100 more mutations in our genome than our parents had. Yet most of the mutations are not bad enough to kill humans before they've had children and pass their mutations along to their descendants. So natural selection, in a sense, is unable to change that, to stop these accumulating, mostly deleterious mutations. So the genome of the whole human race is deteriorating over time. This is called genetic entropy. And the fact that we have not yet experienced genetic meltdown as a species is simply because the human race is just a few thousand years old. The concept of genetic entropy has also been confirmed in various animal species, such as cheetahs, extinct woolly mammoths, and so on. It's also been seen in ancient flu viruses and other things. Incidentally, this concept of genetic entropy is probably why the lifespan of humans dropped so much after the global flood, as recorded in the Bible. At first, when Adam's children were intermarrying with each other, in the data not shown in this picture, their genes were almost perfect, so they would not get as many diseases like cancer or heart disease or other things. But after the flood, as shown in this diagram, showing starting with the lifespan of Noah, after 1,000 years of accumulated mutations, when Noah's grandchildren began intermarrying, their genomes had deteriorated because of genetic entropy, and their lifespan decreased when their gene pool was reduced. And after another 1,000 years, God prohibited close family members from marrying each other in the law of Moses, because so many mutations had accumulated. As we plot the ages of Noah's descendants from the Bible, we see an exponential decay curve in the lifespans, exactly as genetic science predicts for an inbreeding family with decreasing biological fitness. See the supplement for more links about this. One common evolutionist response to this concept of genetic entropy is that most of the genome must be junk DNA, so that most of the mutations we acquire don't affect our fitness. However, as time goes by, more and more parts of the genome that evolutionists used to consider as junk DNA are found to be important and functional, and a recent study called ENCODE showed that at least 80% of our genome seems to have some function. Mitochondrial Eve and Y-Chromosome Adam. Most DNA is inherited from a combination of a father's DNA and the mother's DNA, but mitochondrial DNA in our cells is inherited only from the mother in most cases, and Y-chromosome DNA in males is inherited only from fathers. It turns out that when scientists compared the sequenced mitochondrial DNA of parents and children and measured how many mutations had occurred, they discovered that everyone on Earth descends from a single woman. They labeled this woman Mitochondrial Eve. The average human is only about 22 mutations away from the consensus Eve sequence of mitochondrial DNA, and even the most mutated individuals are only about 100 mutations away from this consensus sequence. The direct pedigree mutation rate in human mitochondrial DNA is measured to be about 1 mutation every other generation. So getting 100 mutations distance would only take about 200 generations, which would be about 4,000 or 5,000 years. So the biblical timeline fits very well with this. Of course, the evolutionary theoretical dating puts mitochondrial Eve around 150,000 years ago, but the actual data does not need that much time. Also, separately, other scientists had studied the Y-chromosome mutations passed down from fathers to sons, and they measured the direct pedigree mutation rate as about 3 to 5 mutations per chromosome per generation, based on the latest high coverage studies. Creation scientists Carter and Sanford also observed that most human men are somewhere around 300 mutations away from a consensus sequence, which is commonly labeled Y-chromosome Adam, the man that all modern men descend from in the Y-chromosome. So at the measured mutation rates, all the Y-chromosome differences we see today could have been generated within 6,000 years, and likely within even less, within 4,500 years. Of course, the evolutionists date this person to hundreds of thousands of years ago. The Bible indicates that all men today are descended from Noah, who lived about 4,500 years ago, and there could well have been more mutations accumulating in the past due to fathers being older when they had their first child, and several other factors discussed in the supplemental links. So again, the biblical timeline fits very well with this scientific genetic data. As Carter and colleagues said about the Y-chromosome DNA and mitochondrial DNA, they show that a single primary dispersion of humanity in the recent past, multiple major branches have arisen from surprisingly closely related individuals in very short windows of time. The surprisingly similar DNA between modern people has caused some evolutionists to theorize that there must have been a recent bottleneck or near extinction event, where the population size of evolving hominids in Africa dropped from hundreds of thousands down to only 10,000 or even 2,000 individuals, and perhaps stayed at a low population size for a long time, of even up to 100,000 years, to reduce the genetic variability and linkage disequilibrium down to what we measure today. While the dates they suggest, such as 20,000 years ago, are different than the Bible's recent creation of the count, it is interesting that they recognize the genetic evidence we see in human DNA today must have come from a small population of humans in the recent past, because we are so similar to each other genetically. There's so much more we can investigate, such as how the haplogroup frequencies line up very well with the biblical accounts, how the Y-chromosome branches seem to match very well with recorded historical migration patterns when the biblical timescale is used, and much more. Here are 3 helpful books, and see the supplemental links for more details, and see also these 2 websites for great details.